Hi everyone. For chapter 10, we will discuss the anatomy and physiology of our muscular system. So just like the bone chapter, we will not be going over the identification part in this chapter, but the basic structural anatomy microscopically, the chemical mechanism of what produces a muscle contraction, and the basic physiology of muscle movements. Now, we will do the muscle identification in the muscle lab, which is what chapter 11 in your textbook goes over as well. So it's not that we skipped over chapter 11, but it's all about identification that we will address in lab instead. And even though we are addressing identification of muscles, we're not actually requiring all the muscles that's discussed in your textbook and your lab manual to be on the lab exercises. Okay, we already narrowed it down to a good list of muscles that you have to know, but you don't have to know every single muscles that's listed in your lab manual. It's just those that I listed in the lab exercises. Okay, so this chapter is definitely one of the harder chapters of the whole course. I would say this chapter and the nervous system, the nervous tissue chapter. So chapter 10 and chapter 12, because it has to do with action potential. And usually students have the toughest time understanding action potentials and all that chemical things that are going on that you have to know in order for an action potential to be stimulated and propagate it and then, transfer onto a muscle fiber. So be sure to pay well attention. Make sure you take your time. Really need to spend the time on this chapter. It's a tough chapter, but students get through it. Okay, if you spend the time to go through all my lecture videos, I try to explain it as clear as I can. And if you're still confused, always feel free to set up an appointment with me. Okay. So, because this is a hard chapter, this is why I have scheduled 2 days for this chapter. It's a lot of information and a lot of complex little parts, okay? So that's why I have that extra assignment for you with the review section of it to help you narrow, at least get through some of the more itty-bitty parts, detail parts down. Okay, so let's get started. The muscular tissues have 4 common properties that we will discuss in this chapter. They're about to be, so for all of them, all muscle fibers are able to be excited upon receiving a stimulus in form of an action potential and neurotransmitters, which we will discuss, towards the middle of this chapter. They are contractible, as we have known it, and meaning that they have the ability to shorten. Okay, that's the whole point of these muscle fibers, is that in order to produce a movement, they have to shorten. Okay, so muscles are always pulling. They're not pushing. They're usually pulling for us. And during that pulling process is that contraction that allows us to have power to move something or move our bones. Okay, muscle fibers are extendable, meaning that they have the ability to stretch and lengthen. And lastly, they are elastic. Either after contraction or extension, they have the ability or capability to recoil back to their resting length. Okay. We will be discussing the details of how muscle tissues are able to do these 4 things. Okay. Now, function-wise, muscle tissues does a variety of things that we have mentioned previously. For example, they can produce movement. Tendons will pull they're attaching the muscles to bones, and when muscles contract, they're pulling on the tendons. Tendon pull on the bones to give you movement. They allow us to maintain body posture and position. Supporting soft tissues, for example, all of our organs, our visceral organs, those in our abdominal cavity, for example. There's this big body cavity wall that's made up of all these different abdominal muscles that you have to identify in lab: external oblique, internal oblique, transverse abdominis. All those things are part of the muscles that help to support those soft tissues underneath, especially the abdominal area, because there's no bones in front of it to protect them. So the muscles does a big part of the support of abdominal internal organs. Guard the body entrances and exits. Now, we won't mention too much about this in this semester. A lot of this comes in the next semester when we talk about sphincters. There's lots of sphincters in our body. Sphincters are like little, I usually like to call it little rubber band holes. Okay, it's kind of like rubber bands where they can stretch. But normally speaking, they're, kind of tied around that area. It ties off your stomach, ties off your ureters, ties off your urethra, etc. So they, excuse me, not ureters, but urethra. So those are sphincters that there are allow liquids usually to move through, particular areas. And some of them can be, involuntary. Some of them is voluntary, where you can control the opening or closing of these sphincters. Okay. So that's why a lot of these has to do with digestive and urinary tract that we will talk about next semester when we get to those digestive system and urinary system. We already mentioned in chapter 1, back way from chapter 1, that muscles does the part of maintaining your body temperature. Especially when we're cold, our muscles can undergo contraction through shivering so that it produces heat for the body. And lastly, they do store nutrients. There's, for example, glucose are stored as glycogen molecules in your muscle cells that we'll talk about in the latter part of the chapter. And you also learned that from your previous general biology class. The other thing that muscles are abundant with is amino acids, because there's all these actin and myosin myofilaments inside each muscle fibers. There's lots of them, okay? And even though, technically speaking, we don't want to destroy those for energy's sake, but if we are in, for example, starvation mode, there's always a backup, which is our muscles. Because amino acids can also be turned into energy. We'll talk about those in second semester in the metabolism chapter, how different pathway of metabolism is compensating for each other, or where does our body go for energy, first, second, and last resort kind of deal. So amino acids is usually the last resort, but there is at least something to burn, okay? All right. So we'll start by talking about the anatomical organization of structure in muscle tissue before we dive into the physiology part of the tissue. Now, when we look at a chunk of muscle like this, okay, so this is your arm, okay, and we're zooming into this chunk of muscle, and it looks like this as a whole piece of muscle. Now this forearm muscle here is actually made up of lots and lots of muscle cells, okay? And for muscle cells, we refer to them as muscle fiber, okay? We talked about this in chapter 4 when we're talking about muscles. Muscle cells are not our usual, little scrambled egg looking thing, okay? Our muscle cells are cylindrical, okay? They're long like this, and they have multiple nuclei like that, okay? Multiple nuclei all along that one single muscle fiber, which is technically speaking one muscle cell. So kind of like your skin cells, your layer of skin in the epidermis are composed of a lot of cells within that epidermis layers. For muscles, same thing. Okay, a piece of muscles is made up of a lot of muscle fibers, which is your muscle cells. But it's just that, we have very different shape and characteristics, for muscles that allows them to be functional for muscle contraction. So that's how they're different from skin cells. But we still have lots of them for all that movement we're talking about. So the organizational breakdown of a chunk of muscle all the way down to a muscle fiber goes like this. So first, on the exterior layer of a large piece of muscle, there is a dense, so we're looking at this here. So there is a dense layer of collagen fibers that's separated from nearby tissues and organs called epimysium. Okay, so that's epimysium. Epi, remember that prefix is for outer. Mysium is a layer that surrounds muscle tissue. Okay, and we'll have something similar for neurons too, where we have epineurium, perineurium, endoneurium. For muscles, it's epimysium, epimysium, perimysium, and endomysium. Okay, epi always means most outer. And just like how bones have osteons, remember that bones, they have, in compact bones anyway, there were these, what I call little logs, those circular logs that I said you can pull out and look at the lamellae, the concentric lamellae that makes up that log. Now for muscles, there's something similar. Now we don't call them osteon, they are called fascicles, okay? So each one of these red circles you're seeing here within the big chunk of muscle is a muscle fascicle. Think of it, sounding like a little red popsicle encased within a bag. Okay, so that's what fascicles are. These red cylindrical tubes that makes up your whole muscle piece. Okay, so for fascicle, it smells a lot like fascia. Okay, so don't confuse between fascia and fascicle. Okay, fascia are those connective tissue layers we talked about in chapter 4. Fascicles are the actual cylindrical muscle fibers, okay, within a big piece of muscle. Okay, and surrounding each fascicle, there is another layer called perimysium. Okay, so epimysium surrounds the entire muscle. Perimysium encircles your fascicles. Okay, and I'll draw this out at the end just to review that whole process again. Okay, so now and then each fascicle, this is one fascicle right here. So we're talking about taking one of this and then we are enlarging it to that and looking at one fascicle. Okay, and each fascicle is made up of many muscle fibers. One muscle fiber is one muscle cell. So each one of these circles you're seeing here is a muscle fiber, one muscle cell. And surrounding each muscle fiber right here, so we're taking one of this and then enlarging it to that, then we're looking at one muscle fiber like this. And that muscle fiber is surrounded by another layer called endomysium. Okay, endomysium. So endomysium surrounds muscle fiber. Okay, so endomysium is made up of, multiple, inside of the muscle fiber itself is made up of multiple protein bunches called myofibril. Okay, so myofibril are these little dots here you're seeing. You see all these little dots in here. Maybe I shouldn't use red for this chapter. It's just a lot of red going on. Okay, blue. All right, so every one of those little blue dots here, okay, those are what we call as myofibril, okay? Which is then your actin and myosin. Okay, within each myofibril, you will have that alternating patterns of thin and thick myofilament called actin and myosin, which is what give us that appearance of skeletal muscle fibers having striations. Okay, the fact that we see striations under the microscope is not because they have these lines that are just red. Okay, they are actual protein fibers within the muscle that produces that very unique pattern of dark light, dark light, dark light that gives you those striation pattern. I'll talk about that in a bit. So perimysium, which is what surrounds your fascicle, contains collagen and elastic fibers, okay, and also have some blood vessels going through. And then endomysium contains capillary network. Muscle fibers needs a lot of blood supply because they constantly need oxygen to power all our movement and usage of ATP, and also for making ATP as well. Myosatellite cells also live within the muscle fiber. So you see this cell right here, that is a myosatellite cell. And what myosatellite cells do is actually pretty cool. There are stem cells that repair damaged muscle tissue. A muscle fiber itself, again, does not have the ability to do cell division, just like your bone cells. Okay, osteocytes can't do cell division. Muscle fibers, same thing. They can't do cell division, but there are stem cells around that can help to repair damaged muscle tissue when necessary. Okay, and then there are nerve fibers within there too, that is for controlling the muscle. So if you look at this tiny yellow dot right there within the endomysium, this says axon of the neuron, which means there is a neuron that kind of sits on top of the muscle fiber for innervating and stimulating it for movement. And that's what we're going to focus on. A big chunk of this chapter is how does the neuron is able to turn on a muscle for contraction. Okay, there's a whole process within there. We're going to talk about that, okay, in a bit. All right, so let me draw this with you one more time to break down the different level of organization of a muscle fiber. Okay, so I'm going to go back to my red now. Okay, so you have your big chunk of muscle, okay, big chunk of muscle with a layer around it. And that layer is epimysium. And inside the big chunk of muscle, you have fascicles. Okay, so fascicles. Okay, each one of these circle is a fascicle. Okay, and if we zoom in to that fascicle, okay, then we get one circle of the fascicle, and each fascicle there is another layer around it called perimysium. And you have muscle fibers within each fascicle. Okay, I'm going to say fascicle. Okay, this is a muscle chunk. Okay. And then if we take out one of those muscle fiber, okay, one of those muscle fibers. So now this is a muscle fiber, which is equivalent to one muscle cell. You have another layer around it that protects it called endomysium. Okay, and inside each muscle fiber, there are many of these little dots. And each of those dots are called myofibro, which is basically your actin and myosin. Okay, and this is something you have to, not this, at least this portion of it is not tested on the lab practical, but make sure you know it for lecture exam. This part in the lab practical, you actually have a model that looks like this, that talks about endomysium, myofibro, actin, myosin, etc. So make sure you pay attention to the more detailed structure of the muscle fiber in a bit, okay? All right, so that's the hierarchy or organizational breakdown of a muscle chunk all the way down to a muscle fiber and the layers that surrounds it, and what do we call those different organization little parts, okay? All right. Okay, so we have mentioned from previous chapter that muscles must be attached to bones via tendons or a wide sheet of tendon called aponeurosis. So you have tendons and aponeurosis. So something like this, which is a thin piece of white fibers that attach muscles to the bone, is a tendon. But if you look at the head, for example, the head has a big piece of aponeurosis or tendon-like thing that connects your frontal part of the muscle, okay, your frontal belly of your epicranius, to the back occipital part. Okay, so that whole white part on the forehead, so what I'm talking about is here is a head. Okay, this is the frontal belly of the epicranius, and then you have this white part right there, and then that's the back part of the muscle, and they're connected together by tendons as well. But because it's a big piece of tendon instead of a tiny little piece like a string, now we're talking a big white sheet of tendon, we call that aponeurosis. Okay. So aponeurosis refers to large piece of tendon, and contraction of a muscle pull is what pulls on the attached bone to give you a movement in a result. So if a muscle fiber or a piece of muscle was to ever be disconnected from the tendon, it can contract all it wants, but you're not going to get a movement is because you need the muscle to pull on the bone in order to pull and give you a movement. Okay. Now, we mentioned that there are muscle stem cells called myosatellite cells. Where do they come from and how do they work? There are type of stem cells, but why do all of a sudden there are these stem cells living around it? Now, in contrast with osteogenic cells in bones, osteogenic cells are very ready stem cells that are ready for repairing damage and all that stuff. Myosatellite, the whole understanding of what activate myosatellite cells, unfortunately, is still kind of unknown. Okay, and whether for example, cardiac muscle, do we have stem cells in our muscles for regeneration of cardiac muscles? Because that would be nice if we can save a person that have passed away from had a heart attack, for example. And a lot of times we can't, if they've been out for a long time after the heart attack, their muscle fibers starts dying without oxygen. And when those cardiac muscles die, we cannot revive them. And what if there are particular stem cells that we can grow cardiac muscle fibers and implant those cardiac muscle fibers back to a heart? That is something that, is always undergoing research. Okay. But, it's still kind of unknown and not as easy or straightforward as we think it should. Okay. So, these myosatellite cells are actually pretty interesting because there's a lot of people that can benefit from this type of, if we can control how myosatellite cells work, especially, if you have heard of the disease called muscular dystrophy. Muscular dystrophy is degeneration of muscle fibers and muscle cells in the person, genetically speaking, and it just happens. There's nothing we can do to stop it. So can we put myosatellite cells, stem cells, back into these patients with muscular dystrophy and let them grow new muscle fibers and muscle in order for them to be able to move and do everyday things? Okay, that's always a possibility. So that's all part of tissue regeneration category. So that is why they talk about myosatellite cells and why is it interesting to study them. So where do myosatellite cells come from? So during embryonic development, there are embryonic muscle cells called myoblasts. Okay, so we're talking about all the way inside the mother's womb when we are forming from our ball of cells. Okay, so these myoblasts actually fuses together to form one long cylindrical muscle fiber with multiple nuclei. So this explains why one muscle fiber can have lots of different nuclei together, is because they come from the fusing together of multiple cells. But some of these myoblasts may not fuse, okay, and they can remain in on the muscle fiber as myosatellite cells, just like this. So these are myoblasts, for example. They can fuse together to form your one cylindrical muscle fiber, but some of them remain as stem cells and doesn't differentiate, and they become just attached to the muscle fiber like that to give you these myosatellite cells. Now, when an injury or muscle damage occurs, these myosatellite cells can enlarge, divide, and then fuse with the damaged muscle fibers to assist in the repair. That's pretty much what we know currently, how myosatellite cells work. But how do we trigger these, and how many of these myosatellite cells we have is still under research, okay? Now, we talked about within each muscle fiber, there are lots of protein bundles called myofibrils, like I was saying. And there can be hundreds and thousands of these within one muscle fiber. So that's how much of these protein little fibers we have, okay, within a muscle cell. Now, what we also notice from the muscle tissue histology is that there are these striations when we look at them under the microscope. These striations arise from alternating patterns of your thin filament, thin myofibril, and thick filament, which is your thick myofibril, okay? The thin one is what we know as actin, and the thick part is what we know as myosin. And these are just little protein fibers that we'll talk about them in detailed structure in a bit because we need to know what are their subunits, what are they doing in order to produce a contraction. Now, these myofilaments are arranged in repeating pattern within each functional unit called the sarcomere, okay? So you see these lines right here. So all of those are the striations we see under the microscope like this. The light area is actin, and then the thick area is myosin. Some of it has myosin and actin overlap. We'll talk about that in a bit. But within this very tiny area, you will have lots of little functional unit down called sarcomere. Okay, and I will talk about what forms a sarcomere in a bit. Okay, we're gonna zoom even further into the muscle fiber to talk about every one of those little parts. Okay, but the sliding of these myofilaments, actin and myosin, is what causes a muscle fiber to contract. Now, since a muscle fiber is a cell, there's, of course, plasma membrane, just like any other cell. So the plasma membrane of a muscle fiber is called the sarcolemma. Okay, there's a specific name for plasma membrane of the muscle cell. Okay, so that's called sarcolemma. Very important because this part comes into critical play of how the action potential gets across a synaptic cleft into the muscle fiber. Okay, so sarcolemma plays a role in that. And then the cytoplasm of a muscle fiber is called the sarcoplasm. Okay, so you can tell already that a lot of things that have to do with muscle starts with sarco. Okay, so you have sarcomere, sarcolemma, sarcoplasm, sarcoplasmic reticulum. So make sure you don't confuse each of these together. Okay, sarcomere is a functional unit of the muscle fiber. Okay, sarcolemma. Okay, remember lemma sounds kind of like plasma. Okay, so it has to do with the membrane. Okay, sarcolemma. Sarcoplasm, just like cytoplasm, this is the cytoplasm of the muscle fiber. Okay, all right. So the sarcolemma, which has a membrane potential, okay, we'll talk about more about this in chapter 12, but just understanding at some sort of electronic voltage due to the presence of specific ions. You should have, your professor from general biology should have slightly talked about it when you were talking about the plasma membrane in that chapter of general biology, but we're going to review that in chapter 12. For now, just understand it as some sort of electronic voltage. Okay, maybe you're at, usually within the cytoplasm, it's more negatively charged, and that negative charge within the cytoplasm is the membrane potential. Okay, all right, so this is what an important feature that will stimulate a muscle contraction when we change the membrane potential. Okay, we're going to get to those itty bitty gritty details step by step of that process in a bit. Okay, all right, so remember the membrane potential will be an important concept, not necessarily in this chapter, but definitely in chapter 12 when we tie the whole story together. This chapter, we're focusing on what's happening within the muscle fiber, which we're not going to focus on those ion movements and electronic voltage going on. Okay, but when we get to chapter 12, then we're going to focus in on those membrane potential and ion channels and all that stuff, okay, to wrap up the whole story. Okay, all right. Or else we try to explain both both concepts at the same time, it gets too overwhelming and too confusing. Okay, so we're just going to talk about the muscle fiber first. Okay, but before we get into that whole process of what causes muscle contraction, we need to know what are the microscopic structures within a muscle fiber that enables it to contract. Okay, so we're going to go over each of these little parts together. Okay, these little parts sounds confused. It sounds like nothing you've known before. So just remember as what they look like. Okay, and then we'll see how they come into play of a muscle contraction when we get to the processes. Okay, all right. So T-tubule, or the long word for it is called transverse tubule. T-tubule are what you see as these yellow tubes right here or right here that are, sort of going vertically around each myofibril. Okay, remember this itself as a whole here is a muscle fiber. Okay, so this is a muscle fiber that we're looking at. And one of these pink cylindrical thing is a myofibril. And what goes around each of these myofibril as these yellow tubes are the T-tubules, okay? And they are sort of that narrow tube that are continuous with the sarcolemma, the plasma membrane of the muscle fiber, and extend into the cytoplasm, okay? Which is the sarcoplasm for the muscle fiber. So you see that this right here is the sarcoplasm, the nude color background, and then you have your sarcolemma, which is that line right there that goes around. Okay, it dips into the T-tubules, which then goes towards each of those myofibrils and into those blue things that we'll know what they're called in a bit. All right, so T-tubule is what makes them an important feature for conducting electronic signal to the entire muscle fiber together, because they are these tubes that goes around all the myofibrils. Okay, it's kind of like the electronic wiring that goes around the entire muscle fiber, that once you have a spark somewhere, okay, that spark is going to be translated throughout the entire muscle very, very quickly in the blink of an eye to tell the whole muscle fiber to contract all together at once. Every one of those sarcomere that we'll talk about. Okay, and you know what I mean in a bit. Okay. So the sarcoplasmic reticulum, so it sounds like endoplasmic reticulum, but don't confuse it with that. They have very different function. The sarcoplasmic reticulum is what forms that tubular network around each myofibril in these blue area right here. So all of these blue coral looking things that you're seeing, or meshy looking things you're seeing, it's like a wrapping that goes around each myofibril, and their job is not to, it's not the electronic part, okay, because that's the T-tubule's job. Sarcoplasmic reticulum is what stores an important ion called the calcium ion, okay? And calcium ion is very critical ion for triggering of muscle contraction. So that's why you need the sarcoplasmic reticulum to wrap around each muscle fiber to release those ions to trigger the muscle fiber to contract, okay? But they will receive the electronic signal from the T-tubule that will trigger the release of calcium ions, okay? So that expanded region you're seeing here, okay, this is still part of the sarcoplasmic reticulum, but there's a special name for it called the terminal cisternae, okay? Terminal cisternae is not a separate part of the muscle fiber structure per se. It's still continuous. It's part of the sarcoplasmic reticulum, but it's a specialized region which is at the ends, either this end or this end, and it's always close to the T-tubule, because terminal cisternae is what's going to be receiving that electronic signal coming from your T-tubule to release calcium ions. Okay, so terminal cisternae here, and then this is another terminal cisternae before becoming your sarcoplasmic reticulum again. Okay, so it's kind of that T-tubule, okay? And then you have the chunky portion of the terminal cisternae right there. And then it becomes the thin portion of that sarcoplasmic reticulum. Same thing on the other side, okay? So this is the sarcoplasmic reticulum. This is the terminal cisternae. Okay, and then this right here is the T-tubule. Okay, so that's exactly what is happening within here. And this whole part with the terminal cisternae, T-tubule, terminal cisternae, this 3 sections of the muscle fiber is known as the triad. Okay, triad, because it's made up of 3 cylindrical tubular things. Okay, that's why it's called a triad. Okay. All right, so and again, the whole thing here is one muscle fiber that we're seeing, and one of these pink tube is a myofibril, and they're made up of myofilaments called actin and myosin that we have already mentioned. So the model you see in lab representing a muscle fiber looks just like this, and that's what it's referring to. Okay, so it's good that you went over this, okay, this part of the lecture. But, if you didn't start early and then you start working on that lab exercise, you might be completely lost. So that's why I have included some YouTube videos from other places within the lab exercise that goes over that model. Okay, I would say this is probably the most confusing model, the muscle fiber models compared to the other models. Okay, so make sure you try to understand this part so that it makes the labeling make a lot more sense when you get to lab exercise on the microscopic view of the muscle fiber. Okay. All right. Okay. So we have mentioned sarcomeres. So now it's time to talk about what makes up a sarcomere. Where does a sarcomere begin and where does it end and what does it have? Okay. So we magnify the myofilaments even further, like this picture right here. So now we're even zooming in even further. So we're taking out one of these myofibril, okay, and then we're taking just this portion of the myofibril and then enlarging it to look at all the different parts of that sarcomere. Okay, so within the magnified parts of the sarcomere, okay, we see these 2 zigzag lines that goes like this. Okay, so those zigzag lines are called Z line or Z disc. Either way is fine. Okay, it seems like the books and everywhere else kind of use lines and disc and zones and bands all interchangeably. So as long as you get the first, at least get the A, H, I, and the Z right, then you're good. I don't care what you put off after that. You can put line, band, disc, zone. I feel like it's very confusing because multiple places will call it different things. So to me, what's going to be critical is the first letter, that big letter. Okay, it doesn't matter if you put band, line, disc, or zone on the lab practical. Okay, as long as you get the first letter, that's good with me. Okay, so from one Z disc, one Z disc to the other Z disc is what we call as one sarcomere, one functional unit of the muscle fiber. So all along the entire myofibril, one myofibril can contain anywhere up to 10,000 of these sarcomeres all connected together. Okay, so this one long myofibril, for example, can have, sarcomere, 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 sarcomere. Okay, that's what I mean by having one sarcomere connected to each other. Okay, so from Z disc to Z disc is one sarcomere, and one long myofibril can have 10,000 of these sarcomeres all connected to one another. Okay, so that's why there's that importance of the sarcolemma propagating that signal very, very quickly, because we got to get all the signal for all 10,000 of these sarcomere within each myofibril, and all the myofibril all together have to contract at the same time. Okay, you're talking about telling millions of people to do a human wave all together at the same time, and that's very difficult if we think about it in terms of human waves. Okay, so but the muscle fiber with all these T-tubule, sarcoplasmic reticulum, they can do it with a snap at the blink of an eye. Okay, that you can tell all those little things to come and contract together, which results in a whole muscle fiber contraction. Because if you have something going out of sync, then you won't have that one nice muscle contraction from the muscle fiber. Okay, so that's why it is very cool how the muscle fiber is able to do this and regulate and control all of these little parts to be contracting together at the same time. Okay. All right, so the striations we see on the microscope is a result of what's called A bands and I bands, which is technically speaking your thick filament and thin filament. Okay, so this right here is your A band. This right here is your I band. Okay, now A band represent where myosin is. Well, some of it is overlap with actin, and because myosins are actually thicker, that's why it appears darker on the microscope to give you those striation lines. I bands are here, like I was talking about, and which are only contains actin. You see those red lines, okay, all along, not the green ones now. The red lines are the actin, okay, and the purple lines here are the myosin, okay. I bands only contain actin, and the green lines called titin, okay, which is a different protein fiber that we'll talk about the function of it in a bit. Now, why we call I band the thin filament is because actin is usually thinner in appearance compared to myosin. So that's why you see them as a lighter color on the microscope. And that's what results in that dark light pattern. So it goes down to dark, light pattern is because you have alternating patterns of A band, I band, A band, I band, A band, I band. Okay, and that's what represents those striations. Okay, all right, so let's break all these parts of the sarcomere down. We kind of talk about A band, I band, Z line. What about the other stuff that we're seeing? What are they doing? What is their role? Okay, and what are they? So let's break all these parts down, okay, which is kind of analogous to that yellow-gray color model you see in the lab of I call it the very high magnified muscle fiber model. So in lab, you will see that there are 3 models for the muscle fiber. One that is a low magnification. I called it the low magnification muscle fiber, and then high magnification muscle fiber, and then the very high magnification of the muscle fiber. There's 3 models, okay? So when we're talking about I band, A band, Z disc, all that stuff, sarcomere, I'm referring to that very high magnified muscle fiber model. Okay, it'll be more clear when you have listened to this part of the lecture and then go to lab, or the other way around. Either way, but you need multiple exposure to it in order to know what I'm talking about. Okay. All right. So the A band contain regions of overlapping thick filament and thin filament. So A band is this, like I was saying, this whole thing right here, and you see blue lines and you see red lines sometimes overlapping with the blue line. Okay, so that overlap we'll talk about, okay, because that is going to be an important feature of why our muscle fibers can contract. Okay, so A band can contain some just only myofilaments, or it can contain both. The M line is the center of the A band. Okay, so these are proteins that connect your central portion of each thick filament to the adjacent thick filament. Okay, so this right here is the M line. Okay, the M line. So M line down the middle. Okay, and then you have your myosin thick filament. Okay. So this is the M line, and it connects these, okay, this side and this side, those myosin or thick filament together to that M line. That's what it's referring to, okay? H band is the lighter region within that, right directly next to the M line that has no thin filament. Okay, I was saying that there are blue and red line right here within the sarcomere, within the A band. Oh, excuse me. Okay, so let me go back to this. Okay, so the H band is this region right here that has no red line as you're seeing it, okay? Here there is a red line, there is a blue line, okay? So those are still part of the A band, but there's a specific name for that area that has no actin, okay? No actin or no thin filament. That's called the H band or the H zone. Like I was saying, kind of keeps going back and forth between those terminologies. And then you have what's called zones of overlap. Okay, so these three things are all part of that A band region. Okay, M line being the middle, M for middle, H band or H zone for that area with no actin, on either side of the M line. Zones of overlap is where you have both the actin filament, the red line, and the blue line together. Okay, zones of overlap. Now, why we have to talk about these parts is because how you can tell a muscle fiber is undergoing contraction is by how much of those zones of overlap you're seeing. Okay. All right. And then I bands. Okay. So this whole thing is the I band. And I band has only thin filament, no thick filament. It extends from A band of 1, because this is the A band of the previous sarcomere. So it extends from the end of that A band to the beginning of the next A band on the next sarcomere. And within the I band, there's a couple things. Okay, you have the Z line, which is what bisects, meaning halving the I bands. Okay, and it marks the boundary between adjacent sarcomere. Remember I said from Z disc to Z disc or Z line is one sarcomere. Titin is those green curvy lines you're seeing. Okay, so these green curvy line is a titin, and same thing here. Okay, that's what this is referring to. So what it does, it extends the tips of your thick filament, which is your myosin, to attachment sites at the Z line. So you see how at the end of your thick filament right there, you have that green line that attaches it toward the Z line. Now, this is very critical is because that's what holds our muscle fiber together. Titin does that very important job of recoil, okay, allowing our muscles to recoil back. So, for example, once your muscle fiber has contracted, your sarcomere needs to go back to their resting length. And in order to do that, your titin can recoil it back so that you have that resting length, and then you can go for another round of contraction. The other thing titin does for us is that it helps to resist extreme stretching, okay? So that we don't pull and damage our muscle fibers apart. We talked about how our muscle fibers are very critical and they may or may not be repaired, okay? So that's why, we don't want to stretch out our muscle fibers so much that all these little lines, A bands, I bands we're talking about starts falling apart from each other, because once they fall apart, then you have no muscle fiber contraction. So that's what two things that titin does for us. One is to help with recoiling. Second is to make sure that your muscle fiber does not overstretch. Okay, so kind of like what a spring does. Okay, so it's like a little spring at the end of your thick filament that kind of makes sure that if you're contracting it this way, it's going to pull it back that way. And if you're stretching it too much, it's going to stop it from stretching it too far down. Okay. All right, so that's all the parts, nitty-gritty parts of the sarcomere. Okay, make sure you are referring this to the lab manual, a lab exercise model where you have that gray, yellow, color model. Okay, so that's what it's referring to, those M line, H zone, etc. Okay, all right, so I'll re-emphasize it here again. So M line, thick filament, thick filament, thick filament. And then you have thin filament attached, thin filament. So this area where there's no thin filament is H zone. Okay. This is your thick filament. This is your thin filament. Okay. And then your entire thing going from the dark to dark is your A band. And then when it was just light thin filament, that's your I band. okay? And then wherever you have both thick and thin filament together, that's called zones of overlap, okay? All right. Now we will zoom even further, okay, even further into the individual protein component of the myofilament structures, okay? This is normal. I usually spend the first lecture in the classroom just going through the structural names of the muscle fiber, so everyone's clear before we get to the physiology processes, okay? Okay, so we'll start with actin first, which is the more complex protein structure compared to myosin, okay? So actin, or the so-called thin filament, is made up of 4 main proteins: F-actin, nebulin, tropomyosin, and troponin. And for F-actin, I will refer them to more so G-actin, because F-actin is the entire string of balls you're seeing here, okay? So 2 strings of balls that are twisted about each other. That whole thing is called the F-actin. But what's more critical are those little individual balls that are making up your F-actin. And each of those little individual balls are called G-actin, okay? All right, so bear with me as we go through each of these components one by one, because they're all critical for the entire process of muscle contraction to occur in terms of cross-bridge formation, power stroke, etc. that we're going to get to, okay? So this is just a protein pulling part. We'll get to the electronic action potential part in a bit. All right. So like I was saying, F-actin is that 2 string of red spheres you're seeing here in this entire actin filament. So remember we were just looking at this as part of the sarcomere. We said the red line is the actin, and now we're zooming in to that red line, looking at this twisted looking thing. That's the actin filament, and then we're zooming in to that twisted thing to look at the individual parts that makes up the actin filament. The red balls twisted about each other is the whole thing itself is called F-actin, F for filamentous, okay? G-actin is the actual individual balls that makes up your F-actin, okay? All right, and what happens is that F-actin is actually twisted around a thin, long strand that you're seeing here in blue right there. That's called the nebulin, okay? So that's what the F-actin is winding around to give you that nice twisted structure of the actin. Each of those red spheres that make up the F-actin is called the G-actin, okay? And G-actin have specific active site for myosin heads to bind, kind of like enzymes, except that they are not catalyzing any chemical reaction. That particular active site is only specific to binding of myosin heads, okay? So these yellow spots you're seeing right there a little bit, okay? You can see them a little bit right there, active site. You see that yellow spot within the red ball is what where the active sites are, okay? Okay, and then tropomyosin are these 2 green twisted protein strings you're seeing here, okay? So that's tropomyosin, and they're responsible for covering up all the active sites on G-actin unless it is triggered by a stimulus to reveal those active sites, okay? Troponin is that brown thing troponin, that brown thing right here that sits on top of tropomyosin, that is responsible for rolling away the tropomyosin to reveal those active sites on G-actin, okay? If you're very confused at this point, it's completely normal, okay? I'll show an animation video in the latter part of the chapter that goes through all these little parts and why all these little parts is important, okay? Like I said, bear with me on talking about these things. because I need to talk about what they are before I can talk about what they do, okay? Now, the troponin itself, as you can see, is made up of 3 subunits. There's 2 circles and 1 long cylindrical part, okay? So those 3 subunits, you have one that binds to tropomyosin, which is the green twisted strings going around the actin, and then you have another circle that binds to G-actin itself to cover up that to sort of just make sure it goes over the F-actin, and one that has a receptor site that binds to specifically calcium ions. So this is why calcium ions is critical part of muscle contraction, because once troponin sees calcium, binds to calcium, it triggers tropomyosin to roll away to reveal your active sites on the G-actin, which then allows for myosin heads to bind and pull in order to produce a contraction. Okay, like I said, it sounds very confusing, but I will show an animation that will go through all the steps to clarify the whole story one more time. And I'm going to draw this one more time as well, okay? Now, at resting potential, when the muscle fiber is not contracting, the binding site on the troponin should be empty, because calcium ion is very low in concentration when a muscle fiber shouldn't contract. We'll talk about what will trigger the release of calcium ions into the sarcoplasm in a bit, okay? And finally, we have the thin filament, which are attached to Z lines or your Z disc at either end of the sarcomere for, right here. So you see this whole actin filament, and then is attached to that Z disc or Z line. Either way you can have it. Make sure the sarcomere stays together. Okay, so let me wrap it up in a nutshell. Okay, in drawing. Okay, so you have one long line first, which is nebulin. And then you have these balls that twist around that line. Okay, this way and the other way. Okay, I'm drawing them as curls, but it's hard for me to draw all those circles one by one. So those are actually the circles, which is made up of G-actin. Okay, and G-actin, if we look at it in zoom in form, it's a big sphere with a particular active site. Okay, that's the active sites. Okay, and this active site is what allows myosin head to bind. Okay, so it's for myosin head to bind. Okay, so that's what that yellow spot on the red ball is illustrating in the picture. And then around the G-actin, you have, I'm going to use another color. Let's use green like it was showing. We have this twisted band that also goes around all the G-actin. Okay, that is your tropomyosin. Okay, what tropomyosin does is that it kind of lays over that active site to block it up. Okay, so that line there, we zoom in to that line, we're looking at this. Okay, we're looking at this where that green tropomyosin line blocks the active site. So myosin head can't bind if troponin remains in that position. Well, what about when we need muscle contraction to happen? What rolls away the tropomyosin? What would take it away? Here is when your troponin comes in. I'm going to use blue, okay? So troponin is 3 subunit. You have one circle here, one circle here, and one long part here, okay? The long part is what right here is what binds to your tropomyosin. One here is for binding to G-actin. One here is for binding to calcium. Okay, so again, this is troponin. Okay, I'm gonna make a zoom in view of it just like what I did there. Okay, one circle, and one thing. Okay, all right, so this is for G-actin binding. This is for calcium ion binding. This is what binds to tropomyosin. Okay, so this is troponin. Okay, All right, so that's the different parts I was talking about. And how troponin is able to roll away tropomyosin is because it is attached like this. And when it binds to calcium, a calcium ion floats in and then binds to one of those sites. It causes this troponin to roll away and pull away the tropomyosin, which then rolls away this green part to reveal the active site that's underneath, allowing myosin head to bind and allowing contraction to happen. Okay, so that's all the parts of the actin filament that's critical for muscle contraction. Okay, and now the thick filament. So the thick filament, which is myosin, on the other hand, is a lot simpler. Okay, not so many little parts. Okay, one thick filament can contain up to 300 myosin proteins bound together in a bundle that looks just like this. Okay, let me change back to red. Okay, just like this in a bundle like that. The myosin head here, right here, is not bound. Okay, see all these little nubs coming off of the thick filament? That's all the myosin head that makes up one of these myosin fiber. Okay, so the myosin head is normally in a resting muscle fiber, is not bound, and they are free to move. Okay, so it swings in one direction this way. Okay, so once it's bound, okay, it can swing in that direction and then goes right back to its original position. And that swinging motion from one side to the other side, okay, so it kind of swings in that motion and then comes right back. Okay, swing in this motion, comes right back, swing in this motion, comes right back, kind of like a little pulling power machine thing. Okay, and the myosin head is what binds to those active sites on G-actin and pulls the actin towards the middle. Okay, again, you'll see what I mean when I show you that video. Okay, so all myosins are arranged with tails pointing towards your M line. So these are your myosin tails, and then all of these nubs here are your myosin heads that are responsible for binding to actin. H-band is what includes the central region of the sarcomere right here, which does not have any myosin head. So that's why there's no actin filament there. Actin filament has to bind to the myosin head in order for it to do its job. So that's why here, where there's no myosin head, there's no thin filament. That's why you have that H-band region where it doesn't have any thin filament. And then each thick filament has a core of titin at the end like this. Remember I said those coil of springs that does two things. One is for recoiling of the muscle fiber back to resting length after contraction. And if a muscle extends, and make sure it doesn't overstretches the muscle because titin is there to pull on it. Okay? And then on the other side of the M line, you have another thin titin. So you have titin here, and then you have a titin there to make sure that everything stays together in one piece as one sarcomere. And many sarcomere is what makes up your myofibril, and many myofibril is what makes up one muscle fiber. Okay? All right. So now that we understand the detailed structure of thin and thick filament, how do they produce a muscle contraction together? Okay, so this is known as the sliding filament theory. I know it's been an hour, but I want to go through the sliding filament, the video animation that goes through all the I-band, A-band stuff, and then we'll take a break there, okay? All right, so the sliding filament theory is when a muscle fiber is in its relaxed state, the zone of overlap should be small. So this right here is showing you a relaxed myofibril, meaning that your muscle fiber is not contracting. Here is what one of those sarcomere looks like. Remember, your one myofibril contains thousands of these sarcomeres all lined up together. Okay, so at relaxed myofibril, you have this zone of overlap that is very small, because what happens when a contraction happens is that this red line needs to climb in and starts moving inwards all of these red lines will then move inwards together, okay? All of these red lines will move in together, okay? So again, the zones of overlap is small, and you have a large H band, okay, because now the actin filament hasn't contracted in yet, okay? So you have all this space for the actin to move into to give you a muscle contraction. Z discs are far apart from each other when the muscle fiber is in that relaxed mode, okay? Now, when a muscle fiber contract, what happens is that the thin filament, these red lines here I was saying, will move inwards towards the M line, okay? Inwards, move inwards, move inwards. These also move inwards. Then the H bands and I bands will now narrow. So this is what's represented a contracted myofibril, okay? A contracted myofibril, you see how this red line now has traveled inwards towards the M line in the middle. Okay, so your H band now is a smaller size compared to this big size H band right there. Your I band is also squished, okay, because now you have that contraction going on. You see how titin is in that compressed spring-like thing now, okay? So titin then will spring back to allow your muscle fiber to recoil back to that resting state, okay? So I band, H bands narrow. Zones of overlap, which then now is widened, okay? You have a tiny zone of overlap right here when it was resting, but once all those actin filament has traveled in, your zones of overlap will widen and increase in size. Z lines are now closer together because Z line goes from here to here now. So the whole sarcomere is contracted and in that smaller length, whereas resting is in that longer length, okay? And the width of A band, however, will always remain constant because remember, it is not the myosin that's doing the movement, it's the actin that's traveling inwards. Myosin heads are always stuck there. They're stuck in that size. That's why A bands never change in length in a muscle contraction. What changes in length is your I band, H zone, and your zones of overlap due to those red lines of actin moving inwards toward the M line, okay? Myosin heads don't move. So this is what we call the sliding filament theory. The sliding of the thin filament towards the M line in each sarcomere is why they call it the sliding filament theory. Okay? And during contraction, sliding occurs in every sarcomere along the myofibril. All the myofibrils then gets shorter, and so does the entire muscle fiber, okay? So every one of these sarcomere you're seeing along the myofibril, they will contract together at the same time, moving towards the M line, 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 and result in this whole muscle fibril, myofibril to be contracted, which then results the entire muscle fiber to contract. And when all the muscle fiber contracts together, your entire piece of muscle will then contract, okay? And in an actual muscle, one end of the muscle is actually attached, okay? We don't have freely moving muscle where it's just sitting there on its own and you're moving inwards from both ends. All of our muscles have two ends, one that is attached and fixed, that is not doing movement, okay? Attached to something. So that will be, for example, your tendon or aponeurosis that is connecting to the bones, for example. That is the origin. Okay, so this fixed end is called the origin. And when every one of those sarcomere you're seeing undergoes this sliding filament theory of contraction, the entire myofibril contracts. Okay, it contracts in one direction towards the origin. And this is what we call the insertion site, okay? That's what's moving towards the origin. When this contracts this way, it pulls on that particular bone. Let's say if this was the humerus, this is the radius or ulnar, okay? And then the elbow is able to move because your muscle fiber is shortening. Your muscle fiber is moving this way. So that's why it pulls on the bone this way to move your arm, move your leg, move whatever it is that you're trying to move. Okay. So that's why for muscles, that's why you had to know origin and insertion sites, as I have put in your lab exercise. I already simplified that down. Okay. I only put the bone names. I didn't ask you to know the processes and exactly where is attached to. I figured that's already too much. So I'm already simplifying the insertion and origin sites down to just simple bone names rather than bone markings. Okay. Okay, so this is that video I was talking about that should go over the review with you, that hierarchy of, endomysium, epimysium, perimysium, and the fascicle and the myofibril, all that stuff, more in a three-dimensional animation style. And then we're going to get to the physiology part of the chemical process, and then another video that talks about all that I was talking about with troponin, tropomyosin, calcium ion binding, etc. Okay, so let's listen to this video.
Skeletal muscles, like the biceps brachii, attach to bone via connective tissue called tendons. Muscles are composed of bundles of muscle fibers. Each bundle is separated by connective tissues known as perimysium. Each fasciculus is made up of muscle fibers, which are separated by connective tissue called endomysium. Skeletal muscle fibers or cells are multinucleated and striated in appearance. Muscle cells are composed of subunits called myofibrils. Each myofibril is made up of several myofilaments. The two types of myofilaments shown in red are composed primarily of the protein myosin, and a thin myofilament shown in blue composed mainly of the protein actin. The repeating arrangement of thick and thin myofilaments serves as the fundamental subunit of striated muscle contraction. These subunits are called sarcomeres. A sarcomere contraction is represented by the shortening of the distance between the Z lines. The sarcomere shortens because the thin filaments slide past the thick filaments. In 3D, each thick myofilament is surrounded by 6 thin myofilaments arranged in a hexagonal pattern. The 3D arrangement of sliding myofilaments is the microscopic basis of muscle contraction.
Okay, and that's what I was talking about, how you have an origin right there, where that biceps brachii is attached to the humerus, and then you have an insertion site right there by that particular tendon. And when this whole muscle fiber shortens is what allows your forearm to move upwards, is because of the shortening of the entire muscle, which is all made of tiny little muscle fiber with all those thousands and thousands of those sarcomere coming together, all shortening at the same time to give you that muscle contraction. Okay, so let's take a break here and we'll come back in part 2 of the chapter 10. And like I said, I think this will be another chapter where we're going to break it into 3 parts, just like the tissue chapter, because it's a pretty long chapter.